Hi, welcome to my YouTube channel. Today we are going to look at the concept of chemistry of nucleic acid. My name is Mukhtar Ali Ikara from Department of Biochemistry, Ahmadu Bello University, Zaria. Introduction to nucleic acids. So nucleic acids are biomolecules essential for all known forms of life. This nucleic acid is a biomolecules, and of course it is one of the macromolecules. They are they are large molecules that are found in each and every living organism. So all living organisms contain these nucleic acids, and they are primarily involved in storage and the transmission of genetics information. So all our characters, all our traits like color of skin, intelligence, the two eyes that we have, the two ear, the one nose that we have, the hairs that we have, our two legs, our two hands, they are all due to these nucleic acids. So, and of course, we as a human, we are different from other species of living organisms like goat, like chimpanzees, like sheeps, like monkeys, donkey, all we are different from them because of this genetics material that is DNA. So these characters, the characters we have, the trait that we have that make us different from other species of living organisms is due to these nucleic acids. And of course, the intelligence, the color of our skin, the wholeness that is found in humans, they are all stored in these nucleic acids. And of course, we inherit the characters or traits through this genetics information. Like if the parent are tall, that tallness is because it is stored in the genetics information, so it can be passed the young ones because generally we inherit the DNA directly from our parents. So if our parents are tall, there is a likelihood that we are going to be tall. If our parents are dull, then there is a likelihood that we are going to be dull. Because intelligence and dullness are also stored in these nucleic acids. So DNA or this nucleic acid, they are very, very important in controlling the characters and traits of each and every living organisms. And they are there stored in the nucleic acids. So generally we have two types of nucleic acids. Deoxyribonucleic acid, which is known as DNA, and ribonucleic acid, which is known as RNA. And of course, this is the structure of the two nucleic acids. So we have DNA. This DNA, if you can see clearly, the DNA is double-stranded. If you look at it, it is double-stranded with larger in sheath. And this is the RNA, and RNA is single-stranded. And one very interesting thing, the DNA and RNA that are made up of four nitrogenous bases. In RNA, we have adenine, uracil, guanine, and cytosine. These are the four nitrogenous bases that are found in RNA: adenine, uracil, guanine, and cytosine. While in DNA, the nitrogenous bases that are found are, we have adenine, we have thymine, we have guanine, and we have cytosine. And as I said very interestingly, ladies and gentlemen, this DNA or this genetics material, which is nucleic acids, they are responsible for controlling each and every character. Anything that you see in humans is because of this nucleic acid, because the nucleic acids is the language of life. We are intelligent because of our DNA. We are beautiful because of our DNA. So anything that you see is because of this nucleic acid, especially DNA. Because in humans, we have uh, different organelles. And these organelles that are found in humans includes nucleus, mitochondria, and chloroplast. So in humans, the DNA are present in the nucleus and in the mitochondria. But the DNA in mitochondria and the DNA of the nucleus differ. And as we continue with the lecture, we are going to look at the differences between the DNA in the nucleus and the DNA in the mitochondria. And of course, one very interesting thing in humans, the human DNA contain about 25,000 genes. So there are about 25,000 genes in humans. There are about 25. There are about 25,000 genes in humans. And each of these genes is responsible for producing a functional protein. Like, for example, in humans, there is a gene called FOXP2. There is a gene called FOXP2, FOXP2. So this FOXP2 is a gene that is there in DNA and is responsible for producing, this FOXP2 is responsible for producing a protein that allow us to talk. So that is why, because in humans there is that FOXP2, and that is why we talk. And of course, there is one bird also that can talk. I think it's parrots. It's also that bird also talk, and it's because it has that FOXP2. And any organism that do not contain FOXP2, it means that that organism cannot talk. So it's because we have that FOXP2 in our DNA, it means that it's stored in our DNA, and of course, it can pass to our younger ones, or it can pass from parent to their offspring. That is why as our parent talk, and you also talk. And if you actually, or if you give birth to a child that cannot talk, it means that the problem is due to some problem that is present in the FOXP2 gene. That is why the child cannot talk. So DNA is the genetics material, and it is very, very important, and is responsible for controlling each and every of our activities. In fact, the DNA is the language of life. You are the way you are because of your DNA. You are beautiful because of your DNA. You are intelligent because of your DNA. You easily get anger because of your DNA. You laugh because of your DNA. You pound your face because of your DNA, because of course, fear and anxiety are controlled by a hormone called adrenaline, and adrenaline is a protein hormone. So it means that there is a gene, because there is a gene in our DNA that produces that adrenaline. So the lower the concentration of the adrenaline you produce in your blood, in the adrenal gland, the lower the anxiety you will develop. So that is why some people are very patient. They easily calm down because their body generally produces this adrenaline in small concentration. While some people can easily get anger because in their DNA they produce high concentration of this adrenaline. And one very interesting thing that we should understand is that some of this DNA, or some of these proteins that are produced by this gene, there are proteins that they can easily destroy, or they can easily degrade it once they elicit their functions. And one of them is adrenaline. And that is why we usually don't get anger throughout our life. So once you produce adrenaline, you get anger. And once it's degraded, then you come back, or you come down to normal. So we should understand that. So as we continue with the lecture, we'll see that the DNA is very, very important in controlling each and every activities of each and every living organism. And it's because of this DNA that is why we are differ from one another. What happened in the case of identical twins? In identical twins, they look alike. They talk the same way, the same shape of nose in some cases, the same shape of face because their DNA is very similar. So we should understand that. So DNA is responsible for storing and the transmission of genetics information. So these are the two nucleic acid, DNA and RNA. Then the next thing is history of nucleic acid, the history of nucleic acids. So the nucleic acid was first discovered by a scientist called Friedrich Mister. So Friedrich Mister in 1869 is the first person that was able to discover the nucleic acids in the year 1869 at the University of Birmingham, Germany. And he named the nucleic acid nuclein. So initially, the nucleic acid are called nuclein, which was later renamed as nucleic acid. But at the initial stage of discovery, the nucleic acid are called nuclein. And the first person that discovered this nucleic acid is Friedrich Mister in the year 1869. And this is the image, or this is the picture of Friedrich Mister, the first scientist that was able or that discovered the nucleic acids. And then the next one is actually Albrecht Kochel. Albert Kochel in the year 1881 is the first scientist that was able to purify nucleic acids. So he's the first scientist that was able to purify and isolate nucleic acids. And at the same time, is the first scientist that was able to discover that the nucleic acid have acidic properties. So you see, this is two important discovery. He's the first person that's purified the nucleic acid, one. So he purified the nucleic acid and is the first scientist that demonstrate the acidic nature of this nucleic acid. And that is why they are now called nucleic acids. He later also identified the nuclear bases, that is the nucleotides that form this nucleic acid. So he actually made a significant discovery in the field of nucleic acid chemistry. He purified the nucleic acids, he demonstrated the acidic nature of nucleic acids, and then he he identified the nucleobases, that is the nitrogenous bases that actually involve in building the nucleic acids. And then later, another scientist in 1889, Richard Altman is the first scientist that created the name nucleic acids. So he's the first scientist that created the name nucleic acid. At that time, DNA and RNA were not differentiated. So nobody at that time knows the differences between DNA and RNA. So they were not differentiated. And that was the time that he created the term nucleic acid, which was still used. Then the next thing is actually another scientist, which is Asbury and Bell in the year 1938, that the first scientists that actually published the X-ray diffraction pattern of DNA and trying to demonstrate the structure of the DNA. Of course, X-ray diffraction pattern is a technique that trying to demonstrate the structure of a biomolecules based on the X-ray scattering. So based on the pattern of the atoms that are present in the DNA, this Asbury and Bell was able to demonstrate the X-ray diffraction pattern of DNA. And then, of course, the history continues. In the year 1944, the Avery McLeod McCarthy experiment showed that DNA is a carrier of genetics information. So they are the first scientists. These are the three scientists, Avery, McLeod, and McCarthy, These are three different scientists that was able to experiment or that was able to demonstrate that DNA is the carrier of genetics information. That is, it is responsible for inheriting character from parent to the from offspring, from parent to their offspring. So these are the three scientists that was able to demonstrate that. And then in addition, in the year 1953, Watson and Crick, they are the scientists that was able to demonstrate the double helix structure of DNA. So this is actually the two scientists, Watson and Crick. If you can now look at it clearly, this is the DNA. It looks like a ladder. So if you look at it clearly, you can see that the DNA is double helix. This is the first helix, and this is the first helix, and then this is the second helix. So this is the double helix structure of DNA. And then the next thing, experimental studies of nucleic acid constitute a major pattern of modern biological and medical research and form a foundation in the fields like. So due to the experimental studies, due to this discovery of DNA, it unraveled a lot of potential in different areas, especially in the area of modern biology and medical research. Let me tell you, ladies and gentlemen, before the discovery of DNA, biology and medical research is not very sound. So discovery of the DNA unraveled or unlocked a lot of application, especially in the field like genome and forensic science. Because before the discovery of DNA or before the discovery of nucleic acid, it is very difficult to understand human genome and the forensic science. But now, due to the discovery of forensic of DNA, you can be able to identify crime using a DNA fingerprinting. So it actually opened up a room in solving a lot of criminal cases. And then second, it is also help a lot in the area like biotechnology and pharmaceutical industries. Like for example, currently due to the discovery of this DNA, scientists was able to demonstrate or was able to produce a recombinant insulin, recombinant oxytocin, which was now used in the treatment of diabetes and also in labor. And of course, in addition, it helps in pharmaceutical industry. So due to discovery of DNA, scientists was able to express a lot of proteins as a target in drug development. So that is why this discovery of the DNA is unlocked a lot of potential in modern biology and medical research in solving a lot of problems that are difficult to be solved. But due to discovery of DNA and or nucleic acid in general, it opened up a room, it opened up a doors in many research, especially in biology and medical research. So now the next thing, what are the types of nucleotides? So nucleotides actually are the building blocks of nucleic acids. So we build this nucleic acid, DNA and RNA are made up of nucleotides. And the nucleotides are also constitutes the three important components. So generally, the nucleic acid are made up of three important components, and you shouldn't forget that. So whenever we said nucleotides, the nucleotides are made up of nucleotides, which are the building blocks, or the blocks that are used to build the nucleic acid are nucleotides. So these nucleotides are made up of three components, and these components include sugar, phosphate group, and nitrogenous base. So it's made up of sugar, phosphate group, and nitrogenous bases. So nitrogenous bases, sugar, and phosphate group are what constitutes the nucleotides. And of course, there are five primary nucleotides. These five nucleotides, we have adenine, we have thymine, we have cytosine, we have uracil, sorry, we have guanine and then uracil. These are the 5 nitrogenous bases. And of course, in addition, these nitrogenous bases are of 2 types. So we have 2 types of bases. So the first basis, we have what you call pyrimidines. And these pyrimidines, we have thymine, we have cytosine. And of course, in addition to thymine and cytosine, there is also a uracil. So uracil is also another examples of pyrimidines. And then in the case of purines, we have adenine and guanine. So adenine and guanine are the 2 examples of purines. So nitrogenous bases are 2. We have 2 types of nitrogenous bases, pyrimidines and purines, and you shouldn't forget that. And pyrimidines is made of thymine, cytosine, and uracil, which is not mentioned here. And then for the purines, we have adenine and guanine. So, of course, a sequence of this nucleotide encode genetics information in DNA and RNA. So the genetics information that we are seeing are there in DNA and RNA are formed by these are formed by these nucleotides. So let's look at the structure of this nucleotide very well. So first we said that in this nucleic acid, DNA and RNA, there are nitrogenous bases. And there are sugar. And this sugar is a pentose sugar. So when you said pentose sugar, it means it's a sugar that have 5 carbons. So let's look at it. We have 1 carbon here. We have 2 carbon here. We have 3 here. And then this is 4. We have 4 here. And then lastly, we have 5. We have 5 here. So this is the 5th carbon. So if you can look at it clearly, we can see that from here, we have 5 carbon. Also, this one is 5 carbon. So what is the difference between this one and this one? So you see, this is a ribose sugar. So in the ribose sugar, look at the carbon 2. So this is carbon 1, and this is the carbon 2. So you can see clearly in carbon 2, there is OH group here. There is OH group, so that is why it is ribose. And the carbon 2 here, in the case of 2-deoxyribose, it is 2-deoxyribose because here there is no oxygen, there is only 1 hydrogen. There is only 1 hydrogen. And here, because there is OH group, that is why it's called ribose. So ladies and gentlemen, I'm going to make this point very clear. Ribose sugar is a ribose, or is a sugar that found in RNA. Ribose sugar, that is the carbon with OH group. In the case of ribose, to ribose is the sugar that are found in RNA. So get this. This is one of the main differences that you should understand whenever it comes to RNA and DNA. Although I mentioned previously that the RNA is single-stranded and DNA is double-stranded. So you should also put that into consideration. And then in the case of DNA, we have deoxyribose sugar. So the 2-deoxyribose sugar is the sugar that is found in DNA. So DNA, remember, is deoxyribonucleic acid. So it's because of these 2 deoxyribose, and RNA is ribonucleic acid, and it's because of the ribose sugar. So then the next thing is the nitrogenous bases. So the nitrogenous bases, these are the 2 structure of the this is the real structure of the nitrogenous bases. So nitrogenous bases is a heterocyclic compound. When we say heterocyclic compound, it means that it's a compound that contain, or that apart from carbon, it also contain other atoms. Like for example, if you look at these nitrogenous bases, there is nitrogen in the ring, and there is a carbon. So that is why it is heterocyclic compound. So adenine here and guanine are the 2 examples of nitrogenous bases. And then we have cytosine, we have uracil and thymine. So thymine, uracil, and cytosine are the pyrimidines. So these are the 2 types of nitrogenous bases, as we mentioned earlier, purines and pyrimidines. So the next thing is actually let's look at the differences between nucleoside and nucleotides. So the nucleosides, we should get this very clear. The nucleosides comprise of a sugar and a base. So a sugar it can be a ribose sugar or deoxyribose and a base. So when you have a sugar and a base, so a combination of sugar and a base is what gives nucleosides. And then in the case of nucleotides, the nucleotides, the nucleotides are made up of phosphorylated nucleoside. It means that apart from the sugar and a base, there is also phosphates. So at least one phosphorus group linked in chains to form a polymers called nucleic acids. So we should understand that a nucleotide is made up of three components, a ribose sugar and a base, while nucleotides is made up of a sugar, a base, and a phosphate group. So that phosphorylated group linked in chain to form polymers, and this polymers is called nucleic acids, that is DNA and RNA. So when you have one sugar, or sorry, one phosphate attached to the nucleoside is called nucleoside monophosphate, and if it is two phosphate, it's called nucleoside diphosphate, and if it is three, then it is nucleotide or nucleoside triphosphates. So that is in the case of nucleotide. So this nucleotide linked with another nucleotide in chain to form a polymers called nucleic acid. So a beta glycosidic bond. So this beta glycosidic bond is a bond that links nitrogenous base to sugar in nucleotides and nucleosides. So a nitrogenous base and sugar are linked together through N-beta glycosidic bond. So how this N-beta glycosidic bond formed? So in purines, the carbon one of sugar bond with nitrogen 9 or base. So because we have the carbon one of sugar bond with nitrogen 9 or base, that is why it is N, because it's linked to nitrogen. That is why it's N. The N here stands for nitrogen. So that is why we have N-beta glycosidic bonds. Then in the case of furimidines, the carbon one of the sugar bond with nitrogen 1 or base. So we should understand the sugar and a nitrogenous base bond through N-beta glycosidic bond. So get this, ladies and gentlemen, it is very, very important. N-beta glycosidic bond is a bond that linked sugar and nitrogenous base. So in the case of furines, the carbon 1 of furines bond with nitrogen 9 of base. While in the case of furimidines, the carbon 1 of the sugar bond with nitrogen 1 of the base. So very interesting, really, phosphodiester bond. So in the case of phosphodiester bond, the phosphodiester bond linked carbon 3 or carbon 4 of hydroxyl group of a sugar, a phosphate. So let's look at this very closely. So you see, this is a sugar, and this is a nitrogenous base. And this nitrogenous base is, of course, we can see here through, you see the carbon one here of furines bond with nitrogen 9 in the case of furines. So you see this is a nitrogen 9 for furines, and this is carbon 1 of the sugar. So you see the linked together like this. That is in N-beta glycosidic bonds. And then in addition, also, in the furimidines, the carbon 9, or sorry, the carbon 1 of the sugar form a bond with N1 of the bases. And this is it. See, N1, this is the sugar. So the same as the nucleosides. But in addition, the carbon 3 or carbon 5, like in this case is a carbon 5 of the sugar linked with the phosphates. So this is a phosphodiester bond. So a phosphodiester bond is formed between carbon 5 and the phosphate. So 1, 2, or 3 phosphate groups can attach. So if it is 1, it is monophosphate. If it is 2, it's diphosphate. So you see here we have 1 phosphate group, and here we have phosphate group. So it means that 2 phosphate group attached to. So this is what happened. So at the 3' or the 5' hydroxyl group, so that is, of course, in the case of the carbon in sugar are also named with asterisk as a prime. So then the next one is the structure of DNA. So DNA is double helix, which composed of 2 strands that wind around each other. And each strand is made of a backbone of a sugar and a phosphate groups linked by phosphodiester bonds. And of course, the nucleotide bases are adenine, thymine, cytosine, and guanine, which pair specifically through hydrogen bond. So generally, adenine and guanine pair through hydrogen bond. So let's look at it here clearly. So these are the sugars. So these are the nitrogenous bases. So you see, if you can see clearly here, you see we have adenine. So this is adenine. This is the color of adenine, and this is thymine. So adenine pair with thymine through 2 hydrogen bond. So 2 hydrogen pair adenine with thymine. You look at it here. So 2 hydrogen bond pair them together. Then cytosine pair with guanine. So look at it. This is cytosine and this is guanine. So cytosine pair with guanine through hydrogen bond. So how many hydrogen bonds are found between cytosine and guanine here is 3. So 3 hydrogen bonds are found there. And in the case of adenine and thymine, is 2 hydrogen bonds that linked them together. And of course, each strand is made up of a backbone of sugar and phosphates groups, and they are linked by phosphodiester bond. So the structure of RNA. RNA is typically single-stranded, but can form secondary structure through base pairing. So RNA is generally single-stranded, but it can fold itself, form a secondary structure by base pairing. Its backbone also consists of sugar and phosphate, but it contains ribose sugar instead of deoxyribose. So you see, this is second differences. You see, the first difference, RNA is single-stranded, and then the second difference is the RNA contain ribose sugar instead of deoxyribose. And in addition, RNA has a basis adenine, uracil, cytosine, and guanine, meaning that the uracil in RNA is replacing thymine in DNA. So it means that RNA contain uracil instead of thymine. It contains ribose sugar instead of deoxyribose. It is single-stranded rather than double-stranded that are found in DNA. And look at the structure here. So these are the base pairs. You see the nucleotide of RNA is cytosine, guanine, adenine, and uracil. While the nucleotides of DNA, you see there is thymine, adenine, guanine, and cytosine. So you see this is thymine and this is uracil here. So that is the difference between the two. And then, of course, this is double-stranded. Look at the DNA is double-stranded and the RNA is single-stranded. So what are the functions of DNA? So DNA serves as a genetic blueprint for all living organisms. So it is genetic blueprints of each and every living organisms. So you are the way you are because of your DNA. You are beautiful because of your DNA. Your child or your children are intelligent because of your DNA. Although intelligence is inherited about 80%, 70 to 80% uh, of intelligence is inherited from the mother. Is inherited from the mother. Due to one, breastfeeding. Second, due to XX chromosome, because it means that in women, if they have XX chromosome, it means that they have complete genome. But in the case of men, where they have XY, so it means that something is missing. That is why it changed from X to Y from evolutionary history. It means that something is missing from here. It's missing. So that is why there are some genes that are missing in men, while for women they have it complete. So because of this breastfeeding, the complete genetic material in the X chromosome and also the mitochondrial DNA that is inherited by the child from the mother. So our mitochondrial DNA is directly inherited from our mother, one. And second, because of breastfeeding. And third, because in the XX chromosome, one of the X chromosomes of the mother is having complete genome, and some of the genes in that part of X chromosome is inherited from, sorry, it contains some DNA that are responsible for controlling intelligence. They are there, and that is why 70 to 80% of intelligence is inherited from the mother. Then in addition, very interesting, in addition, the DNA is responsible for the replication and the transmission of genetics information. So the transmission and the replication of genetics information in women is actually through cellular division. Is through cellular division. So during cellular division, DNA is replicated. When we say DNA replication, it means that one copy of DNA replicates itself into two copies and 2 to 4. So that is the DNA replication, like normal mitosis. So DNA also plays a critical role in encoding proteins through the process of transcription and translation. So generally, this is actually how replication takes place. And for the replication take place, there are ingredients that are needed. There are ingredients that are needed for the replication of the DNA to take place. One, we need DNA polymerase. We have DNA ligase. We have RNA primer. We have DNA primase. And then we need a single-stranded binding protein. So what happened and what is the importance of this enzyme? So the importance of this enzyme, the polymerase enzyme, the DNA polymerase, is the enzymes that involve in adding a single nucleotide. So it is the one that is adding nucleotides. So, but I don't want to actually go deep on this because this is the basic chemistry of macromolecules. So we don't need to go into DNA replication, but you need to understand that there are enzymes that are needed for replication to take place, or there are raw materials that are needed for the replication to take place. And of course, as I said, replication is when you have one DNA is converted or it replicates itself into another molecule of DNA. But it is actually a very complicated mechanism which I don't need to explain it for you at this level. So then what are the functions of RNA? So function of RNA, okay, yeah. So I forget to say something here. So the DNA plays a critical role in encoding proteins through the process of transcription and translation. So generally in molecular biology, we have a process called central dogma of DNA, central dogma of molecular biology. And this concept is actually when you have a DNA, convert itself to RNA. So when DNA is converted into RNA, the process is called transcription. And then when the RNA converted to produce or is translated into protein, when you have DNA translated into protein, so the process is called translation. So DNA to RNA is called transcription. And then RNA to protein is called translation. And of course, if it is the DNA, like for example, if for example DNA convert itself into another DNA molecule, DNA to DNA, the process is called replication. So what happened? As I said, in humans, there are about 25,000 genes. And each of these genes is responsible for producing a particular protein. Like normally in the DNA of human, there are about these 25,000 genes. And each of these genes is responsible for producing a particular protein. And how one gene converted into a protein is through the process of this replication, transcription, and translation. Like for example, if this gene one is a gene that is responsible for producing hemoglobin. So first, this gene one will be converted into RNA hemoglobin, the RNA of the hemoglobin. And then the RNA will now convert itself into the hemoglobin HB through the process called translation. So this is how the central dogma takes place from DNA to RNA, from RNA to proteins. And once you have a proteins, you have a function and structure. Like for example, hemoglobin is the protein that found in the red blood cells, which make the red blood cells to be responsible for transporting oxygen. So that is why understanding the concept of central dogma biology or central dogma of molecular biology and the DNA itself is very, very important. It's very, very vital in modern medical research. So what are the functions of RNA? So RNA is primarily involved in protein synthesis as a messenger between DNA and ribosome. So generally first, when you have your DNA, then the DNA will be converted into RNA. So this process is taking place in the nucleus of a cell. It's taking place in the nucleus of a cell. So after the RNA is produced, it will process, and then later it will be transported to cytoplasm. And in the cytoplasm, we have a ribosome. And then the ribosome in the cytoplasm will now be used, or you will now use the RNA from the DNA, the RNA from the nucleus, and it will now use that RNA as a template for protein synthesis. And that is why we said that RNA is used for protein synthesis. So without wasting much of our time, this is the functions of RNA, and that is how it works. So although we are going to look at the other functions of RNA, although generally we have different types of RNA. We have ribosomal RNA, we have mRNA. mRNA stands for messenger RNA, while tRNA stands for transfer RNA, and rRNA stands for ribosomal RNA. And each of these have a distinct rules in this process. That is the process of protein synthesis. So messenger RNA, transfer RNA, and ribosomal RNA have their distinct functions, have their distinct roles in the process of central dogma of molecular biology, that is from DNA to protein. And in addition, RNA can also have regulatory functions. like influencing gene expression and cellular process. So when you say gene expression, the word gene expression is simple. Also, I just actually describe it. So a gene expression means how the DNA, this is the gene expression from DNA, RNA, and then from RNA to protein. So this is actually the concept of so RNA can also have a regulatory function influencing gene expression. So when we say gene expression is when DNA first is converted into RNA, and from the RNA then is function. Function. So protein, the next thing is function. Why do we say function? Because a protein is what determines the function of each and every cell. Like red blood cells is transporting oxygen. White blood cells is responsible for immune system. And it's all because in the white blood cells, there are proteins that are involved in immunity like antibodies. In addition, the liver cells is involved in detoxification and it's because of the proteins that are there. And all these proteins are produced from the DNA. So that is why all the cellular activities or cellular processes are controlled by the proteins. So gene expression is from DNA to protein. And then cellular processes is the functions that are produced by the proteins in a specific types of cell. So you see, as we said, look at the RNA. So we have different form of RNA. So this is a double-stranded DNA in a nucleus, and then the double-stranded DNA will be converted into messenger RNA, forming on the DNA strand, and then the next thing is the formation of protein. But as I said, it's actually a very complicated process, which I think we are not going to discuss that here. Yeah, we are not going to discuss this one, but you should understand that the most important thing is actually how the DNA is converted into protein. And this is a simple uh, explanation from DNA here, from DNA, then we have the messenger RNA, and from the messenger RNA, and then formation of protein. So DNA replication, I don't also want to talk about this. As I said, DNA replication, when you have one copy of DNA is converted into another copy. So ladies and gentlemen, I'm going to stop here. And if you have any question you can ask, please subscribe and join the YouTube community. Thank you very much.